I'll just introduce Keith and we'll go from there. We have a forum. So it's hey, there he is. So it's my pleasure to introduce Keith Maier today for his presentation for his defense. I custom is to say a few things about students. So I'll start by saying that telling a story about how Keith joined the lab. He rotated in my lab the same year that Samantha Bowman came to the lab, and Sam defended a couple months ago. So Keith is sort of hot on the heels. Sam rotated to my lab and then asked to join my lab, and I had already committed to letting Sam join the lab. So when Keith came to the lab, he actually did some really great work, and also wanted to join the lab, but he actually got his initial selection for what he was doing with rotation. But since I had already committed to another student, I wasn't sure if I could commit to another student because he's in the same year. But Keith's crafty, for those of you who don't know. He's very crafty. And so he actually got myself and John Lai and Steve Almo in a room together, and the plan was to basically the three of us would share Keith. That didn't happen. He basically stayed on in my lab. But it was a good sort of ploy to stick around, and I'm really glad he did. Over the last couple of years, Keith's obviously learned how to do selections in the lab, and I'll tell you about the selection he did that took what the lab was already doing, a selection I had already done, and made it better. But he also picked up all the knowledge on chemical synthesis in the lab. He's really basically been the go-to synthesis guy for making Aptamer's chemically in the lab, synthetically, doing all the conjugations, and been really the knowledge base for a lot of that. So I think for pretty much everybody's Aptamers in the lab, maybe for the stuff Sam was working on, she's sort of doing her own chemistry. He's been the guy to go to, and so when he does leave, we're going to miss him. He's already been disseminating his knowledge to the next generation of folks, and Christine in the lab has really been picking up all of the information. It's great to see sort of this knowledge get passed down. I should probably say something funny about Keith. There's a lot of funny things I can say. I think one of the funniest stories I know, or the earliest sort of amusing stories I know, is great, perfect kind of Keith, for those of you who know him, is that when he was rotating in the lab, he had figured out he could sneak up on people. In particular, he could sneak up on Lindsay and scare the crap out of her. And I guess Lindsay had been watching Tom and Jerry cartoons, and she decided that she would put a bell on Keith so that when he walked around the lab, she could actually hear him. But then it only took Keith about 30 seconds to figure out how to walk around with the string of bells still around his neck, because Keith wouldn't take it off. He left the bells on. He would just sort of shuffle around the lab and still sneak up behind him and scare the crap out of him. The other thing that is always kind of funny that happened in the lab is that Keith and I are roughly about the same size, and we had shirts that were similar color. And so Keith would come up and stand behind something, and everybody in the lab at some point in time thought that he was me. Or what was even funnier was people thought that I was him. I won't tell you what happened. But I'll stop blathering, and I'll give the floor to Keith, who will tell you about what he's been working on over the last couple of weeks.
Thank you, Matt, for that great introduction. And I'm really excited to see all of you guys here today. It's good because I really enjoy talking about my work, so I'm glad that so many of you are going to get to hear about it. So let's just get into it. Today we're going to be talking about separating the WAS from the Chaff, a development of a site-specific Aptamer selection strategy. So just to give you a little overview of what we're going to be talking about today. First I'll give you an introduction into Maier receptor, transferrin receptor, as well as Aptamers in general. Then we'll be talking about the two main portions of my thesis work. First is ligand competition as a selective pressure in selects, and the characterization of the aptamer that we pulled from that selection called WAS. And second, we'll be talking about WAS-mediated inhibition of New World mammarinovirus infection in vitro. And then finally, we're going to wrap it up, and I'll tell you about where we think this body of work is going to propel the field in the future. So working in the Levy lab, one of the main things that we're interested in is targeting cell surface receptors that endocytose into cells. This is advantageous because various cell types have unique receptor expression patterns. So targeting a particular receptor or pairs of receptors can give you site-specific localization. And if you pair that with the fact that many drugs and diagnostics cannot pass the plasma membrane on their own, and any drugs that can would be undesirable because of their lack of specificity. So if you pair receptor targeting with these molecules, what you can get is targeted recognition in a site-specific manner, facilitating the entry of non-permeable molecules into the endosome. And we do that through a process called receptor-mediated endocytosis. So my particular receptor of interest is transferrin receptor, over here, my little guy. It's a type 2 homodimeric glycoprotein, and there are 4 domains within the receptor. The apical domain, which we'll talk about a lot today, the helical, protease-like, and then transmembrane domain. And the natural ligand for this receptor is called transferrin, and its job is to sequester iron in the blood, because free iron can cause the formation of toxic radicals or polymers. So what iron does is what transferrin does is when it binds up iron, it increases its affinity for transferrin receptor and then associates with the receptor at a 2-to-1 ratio, so that one transferrin binds on either side of the transferrin receptor. And this ends up covering both the helical and protease-like domains of the receptor. Transferrin receptor itself is interesting because it's expressed on most cell types throughout the body, making it advantageous for looking at the sort of intrinsic system of endocytosis. And it's also particularly important therapeutically because this receptor has been shown to be upregulated in cancer since those cells have higher nutritional needs. They're expressed by the endothelial cells of the blood-brain barrier, facilitating the transcytosis of transferrin across the blood-brain barrier. And they've also been implicated in viral infection, which we'll be talking about more later. So the life cycle of transferrin receptor, it starts out in the extracellular matrix of the plasma membrane, and then it's endocytosed constitutively, so it doesn't require binding of the ligand for internalization. And when it's endocytosed via plasma-mediated endocytosis, the endosomes mature, thus dropping the pH, and when the pH hits 5.5, transferrin loses its affinity for iron, and therefore loses its affinity for transferrin receptor. The iron is then shuttled out of the endosome, and the endosomes are recycled back to the surface where transferrin and transferrin receptor can be reused. So one of those applications that make transferrin receptor interesting is its implications in viral infections, particularly arenaviruses. So arenaviridae are enveloped viruses, so they have a lipid membrane on the outside. And viruses in general need host cell machinery in order to replicate. So the virus actually needs to be able to gain access to the cytoplasm of a host cell to perpetuate its life cycle. And viruses do this. Viruses express a glycoprotein complex on their surface that, when matured, produces 2 discrete proteins, one of which is glycoprotein 1, which is responsible for recognizing host cell proteins that facilitates endocytosis into the endosome. And the other one is glycoprotein 2, which, when the pH of the endosome drops, glycoprotein 2 is activated and facilitates host endosomal membrane fusion so that it can spill its contents out into the cytoplasm. So of these arenaviridae, there are 2 genuses within that family. The recently discovered reptarenaviruses, which infect snakes, and mammarenaviruses, where each species has its own unique rodent hosts. Of the mammarenaviruses, those are segmented into New World and Old World mammarenaviruses, defined by their geographical segmentation between Africa and South America. And of those New World mammarenaviruses, 5 of them are pathogenic due to having a human cross-reactive GP1. And those 5 viruses are Hunin, Machupo, Guanarito, Sabia, and Chapar. Today we'll be talking about Hunin and Machupo, but all of these viruses are classified as Category A bioterrorism agents by the CDC. They cause severe hemorrhagic fever in humans and have a case mortality rate of roughly 20 to 30%. So the interesting thing is that these viruses actually recognize the apical domain of human transferrin receptor in order to facilitate cell entry. And there are currently no FDA-approved drugs for the treatment of these viruses. The only thing that we do know this far is that some apically binding anti-transferrin receptor antibodies are able to compete with the viruses for binding to transferrin receptor. One of those in particular that we'll be talking more about today is clone MA712. So antibodies, as they've been shown, can actually have function in this system, but there are some limitations of antibodies that should be discussed. First of all, they're somewhat difficult to synthesize through a non-trivial bacterial expression system, which can lead to batch-to-batch variability. And although the popularity of antibody therapeutics in industry these days has greatly reduced the cost of antibody production, it is still not exactly cheap. Another problem with antibodies and proteins in general is that they require chaperones for folding, and that makes them susceptible to thermal and stochastic denaturation. Therefore, if they are unfolded, the refolding into a functional molecule is usually irreversible. And due to the fact that these can be thermally denatured, and you don't want that to happen, distribution of antibody therapeutics requires a cold chain. And while that's fairly trivial in the United States, these mammarinoviruses are endemic to South America, and maintenance of cold chain delivery of therapeutics has actually been shown to have upwards of 50% of the cost per dose for protein therapeutics. But while antibodies still have a lot of advantages, we think that some of these limitations for antibodies could be addressed by using aptamers. So the first thing you might be wondering is, what is an aptamer? So an aptamer is a short, single-stranded RNA or DNA sequence that can fold into a functional binding molecule with high affinity and specificity. Just as an example, here we have the anti-lysozyme aptamer bound to lysozyme. And as you can see, the lysozyme aptamer is a single-stranded piece of RNA that folds up onto itself, creating a scaffold that orients these nucleotides for functional recognition of lysozyme. Aptamers are advantageous because they're easy to chemically synthesize. If your collaborator develops an aptamer, they can email you the sequence. We can synthesize it on the synthesizer in a handful of hours, and it can be deprotected and purified within a couple of days and ready to go. And that leads to the fact that since these are chemicals, there's very minimal batch-to-batch variability, and they're also thermally refoldable. So in the process of making aptamers, we frequently heat these things up to 70 degrees Celsius to unfold them, and when you cool it back to room temperature, they will always refold into the exact same structure. Aptamers are also cool because we can make them nuclease resistant and also decrease their immunogenicity in vivo systems. Compared to DNA and RNA, if we replace the hydrogen or hydroxide group on the ribose sugar of the backbone of the oligonucleotide with either a 2' fluoro or 2' O-methyl group, we can greatly increase their resistance to nuclease degradation, as well as decrease the immunogenicity of these molecules as well. Another thing that we really like about aptamers is that they can be modified at any position, site-specifically, with unnatural nucleotide incorporation. So this, because we synthesize these on a synthesizer, at any point we could put in an unnatural nucleotide that includes the addition of a fluorophore or a chelator for direct imaging applications, cholesterol, lipids, or polyethylene glycol to modify the pharmacokinetic or pharmacodistributed properties of these molecules, as well as alkynes, thiols, and amines for addition of anything else that you might desire. And these molecules are identified through a process called selects. And the first step in selects is creating your naive library. So to do this, instead of synthesizing a discrete sequence, we synthesize a random region, and these usually consist of 30 to 50 nucleotides that have the equal chance of having A, T, C, or G in any position. We then flank those with constant regions for PCR amplification and pool them into a library. And while we're only displaying 5 sequences here, these libraries usually have a diversity of roughly 10 to the 15th individual sequences. But for those of you who like to play around with math, you might be noticing that a 50 nucleotide random region actually has 4 to the 50th permutations, which is actually equivalent to 10 to the 30th sequences. So what's the difference here between the actual diversity and the theoretical diversity? Or why can't we make 10 to the 30th sequences? And to answer that, if you have that 90 nucleotide sequence with a 50 nucleotide random region, that's going to weigh 31 kilograms per mole. If you make 10 to the 30th of those sequences, or 2 times 10 to the 6 moles, a 10 to the 30th library would weigh 65 million kilograms. Just to give you a frame of reference, a blue whale weighs 190,000 kilograms. So to make an entire 10 to the 30th library, you would need the equivalent of 342 blue whales worth of RNA. And that's just not practical. So the way we get around this is by sampling sequence space. So if you look at this structure of lysozyme again, we see that there's a scaffold here orienting these nucleotides which are interacting with the protein. So you could imagine that maybe some of these nucleotides here could be able to vary between different nucleotides. Or maybe this GC pair here could turn into a CG pair, or maybe an AT pair, and you would still have a functional anti-lysozyme aptamer. So what that means is that while this is the sequence of the lysozyme aptamer, we could have multiple homologs of it that would still function similarly. So what we do is if we think of this grid here as all the possible 10 to the 30th sequences you could have in a library, we sample the sequence space within that library, creating our 10 to the 15th, hoping that the sequences that we get in the library are close enough to the real sequence of, let's say, the lysozyme aptamer, that we're actually going to get a functional binding molecule. And so once we have this naive library to work with, we find aptamers within it through a process called selects, which is systematic evolution of ligands by exponential enrichment. And you can do this with both DNA and RNA-based libraries. And it all boils down to three fundamental steps, which is you take your randomized library and you incubate it with your target. And that could be a small molecule, a recombinant protein, an entire cell, or even a whole animal. And then you partition away any sequences that don't bind to your target. You then elute the sequences that bound from your target, and then the rest of the process is just amplifying up the sequences that you recovered for use in another round of selection, because this process isn't perfect on the first round, so you have to do it iteratively before you can end up with an enriched library that has the sequences you desire. So, now that you are all aptamer experts, we're actually going to talk about what I did. And the first part of that process is ligand competition as a selective pressure in selects. And particularly, the bulk of this section is going to be about the thorough characterization of WAS, because as our lab has found, under the leadership of Dr. Lindsley Kelly, is that there are a lot of insufficiently characterized aptamers in the literature. So the first thing that we wanted to do with this paper was to really set out a systematic characterization of this aptamer so that we really understand it before moving on with applications. So when I joined the lab, as Matt mentioned, we already had an aptamer against transferrin receptor called C2min, and he made this aptamer using a naive library composed of 2' fluoropyrimidine. So the 2' group on those oligos on the C's and U's were modified with a fluorine group to increase their nuclease stability and also decrease immunogenicity. And then Matt took that pool and he panned it for 4 rounds against recombinant transferrin receptor, following 1 round with transferrin receptor expressing cells, so that the internalization of transferrin receptor would actually sequester functional molecules. And out of that 5th round, he found an aptamer called C2. Unfortunately, what we quickly learned is that while C2 did really well binding to transferrin receptor in media, if we increase the concentration of transferrin in the media, then we see a decrease in the aptamer's ability to be internalized by cells. And this can be a problem because given the fact that the concentration of transferrin in the blood is 25 micromolar, it severely limits the ability of this aptamer to function in vivo. So this sort of leads into the rationale for my selection. And what we did was we knew that the concentration of transferrin in the blood was 25 micromolar. We also know that these New World mammarinoviruses evolved under that selective pressure and managed to settle on binding to the apical domain in order to facilitate their internalization. So we were wondering, can we use transferrin to drive the selective pressure during an in vitro aptamer selection? Or to put it more visually, Matt's selection selected for everything that bound to transferrin receptor. Can we use the footprint of transferrin to restrict the number of aptamers we get out of the selection, thereby increasing our likelihood of finding an apically binding aptamer that will compete with viruses? So what I did was I picked up Matt's round 5 selection, and I did 4 extra rounds of selection, doing internalization selections in cells, but adding superphysiological levels of transferrin to the medium. So the results of that, we're going to be looking at a lot of FACS plots today, so I'm just going to give you a quick overview there. Basically, the way our internalization assays work is we take fluorescently labeled aptamer, and if that aptamer binds transferrin receptor, it will get internalized with the receptor, leading these cells to be fluorescently labeled. We then analyze it by flow cytometry, and we see that compared to unlabeled cells, cells that have internalized fluorescently labeled aptamers will shift to the right. Conversely, if we have a transferrin receptor aptamer that competes with transferrin for binding to the receptor, then saturation of those cells with transferrin will lead to the inability of that aptamer to be internalized into cells, leading in a decreased amount of fluorescence in those populations. However, if this aptamer does not compete with transferrin, it won't care if the cells are saturated with transferrin, and it will still be able to be internalized into the cells, leading to a shift back up to what we were seeing without competition. So if we look at Matt's round 5 pool, which is the one that he found C2 from, we see that it had good uptake into transferrin receptor expressing cells in media. However, if we add supraphysiological levels of transferrin, we see a complete knockdown of that pool's ability to internalize into cells. After my 4 rounds of selection, we see that all of the rounds still manage to function in the presence of media, but if we add supraphysiological transferrin, we see that the rounds eventually manage to enrich new aptamers that were able to internalize regardless of the presence of that transferrin. So now we have a bit of a fuzzy understanding of what's going on. We have a pool of aptamers that we now know doesn't care about the presence of transferrin, but is still able to label transferrin receptor expressing cells. So the next thing we wanted to do is to dive deeper and look inside of this pool. So we pulled out sequences from that pool and looked at them clonally, and here we're just looking at the sequences of the round 9 pool from the selection. Here we have the constant regions on either side, and if we were looking at the round 0 library, this would all be random, but now you can see that it's been enriched for one predominant family here that has over 55% of the sequences. Interestingly, the second family here are sequences that are related to C2. So despite the fact that it competes with transferrin, it was still able to persist through those rounds of selection. So what we did is we took representative clones from each one of these families and transcribed them individually and tested them again in the same way that we tested the pools. And what we found is that while all of them functioned in just the media on their own, only the clone from family 1 was able to function in the presence of supraphysiological levels of transferrin. But we wanted to look and see, so one of the things that we were interested in is how the selection process actually changed, because halfway through the selection, we sort of changed the rules on these aptamers and added a new selective pressure for the 6th round of selection. So what we did is we did deep sequencing, and then with the help of my friend Kevin, we analyzed those pools, and so we had 10 to the 6 sequences read for each one of the rounds of selection. And what we see here is that if we just look at the selection up to the point where Matt found C2 in round 5, we see that C2-like sequences, which had less than or equal to 5 mutations from the parent C2 sequence, those C2-like sequences had risen to 22% prevalence in the pool. Now, the interesting thing that we see here right now is the fact that if we had not performed this competition selection, family 1-like sequences, the ones that don't compete with transferrin, only had a prevalence of 0.2% in the pool. So that means if we had wanted to find this aptamer just from the selection that Matt did, we would have never been able to find it, no matter how many sequences we pulled out and tested. So after continuing the selection, we saw that the family 1-like sequences dramatically took over the pool, and that led to the corresponding increase in efficacy that we saw with those pools being able to internalize to transferrin receptor expressing cells. So now that we have this family 1 aptamer, we wanted to get a better idea of what it looks like. So we plugged the sequence into a program that we use that predicts secondary structures, and this is the structure that it gave back to us with this program called mFold. And the first thing that we wanted to do was to see if there's a minimal portion of this sequence that can still contribute to binding. So we did this by doing a rational minimization, where basically we used PCR amplification to iteratively truncate this molecule to see when we would lose function. So we started by cutting off this tail here to make minimization construct 1, and we found that this construct was still able to internalize into cells when fluorescently labeled. We then cut off this loop stem here to make min2, and found that this functioned when internalizing, when incubated with cells. We then tried cutting off this atypical UU bulge here to make min3. And interestingly, this one did not function, which is kind of interesting because you don't typically see this type of structure here. And just to make sure that this UU bulge was actually important, we also mutated two of those U's to A's to create min4 construct, and this construct also had severely hampered ability to internalize into cells. So this tells us that min2 is the smallest that we're going to make this aptamer, and after staring at the secondary structure for a while, Matt decided that we should call it WAS, because it kind of looked like that monster from the Monsters Inc. So that's how we got WAS. And now that we have WAS, so we have this sequence that we pulled out of this library, right? But we only sampled 10 to the 15th sequences, so it's possible that WAS isn't a fully optimized aptamer, and it could be better. So what we did was we took the parent sequence of WAS and synthesized a new selection library, where in each position we had a 30% doping, so that there was a 70% chance that the library would have the nucleotide of the parent sequence, and a 10% chance they would have one of the other 3 remaining nucleotides. And that lets us sample the sequence space around this molecule that we had selected. So after turning that into a pool and doing 4 more rounds of competition selection on cells, we found that while the mutagenesis did diminish the functionality of the pool, by the time we'd selected and gone up to round 4, the pool had regained the functionality similar to the F1 clone. And but interestingly, when we pulled sequences out of this round 4 pool, what we found is that none of them really functioned better than WAS. So by some miracle, we had actually found the best iteration of WAS in our first selection, despite the fact that we sampled such a small amount of sequence space. So the next thing that we did was we took the sequences from the doped 4 round, and we submitted those for deep sequencing, and then we mapped all of the mutations found within that pool out of the 1 million reads that we got from that experiment, and we mapped the mutations onto the predicted secondary structure of WAS to see if we could confirm that predicted secondary structure with an experimental method. And what we found is that greater than 95%, all of the nucleotides involved in the structural elements of the aptamer were conserved at greater than 95%. So that means that despite the mutagenesis, all of these base pairs still managed to come back out the other side of the selection, indicating that this probably is the actual structure of this aptamer in solution, at least at a secondary level. And we also got secondary structure confirming mutations. For example, this GU pair was maturated to a much more thermodynamically stable GC pair 37% of the time. And we also saw structure confirming mutations where a CG pair would flip into a GC pair and also be replaced by a TA pair, indicating that the aptamer doesn't really care what nucleotides are there as long as there's base pairing happening. Interestingly, we also assayed some of the more prevalent mutations out of this experiment and found no change in observed affinity. So we really did find the best iteration of WAS after our first pass. So now we have this minimized aptamer, and we've truncated it, we've pulled it out of its pool, so we just want to reaffirm that it still has the characteristics that we saw when we had assessed the pools, or at least competitively. So what we did was we took fluorescently labeled C2 or WAS, and if you compare it against just the aptamer by itself here in red, if you compete C2 with itself, you'll see a knockdown in C2's ability to internalize into cells. And as we had already found out, if you compete C2 with transferrin, you get a knockdown in internalization. However, if you compete it with WAS or C36, which is just a control aptamer that we'll talk about a lot today, you don't see any effect on its ability to internalize. If we take it over to WAS here, we find that compared to the WAS internalization on its own in red, only competing WAS with itself inhibits its ability to internalize into cells. So this is confirming that WAS does not bind with transferrin receptor, but also, interestingly, shows us that C2 and WAS, while both binding transferrin receptor, don't compete with each other, so we could possibly use these for cooperative binding molecules in the future. All right, so now we have a pretty good understanding of the secondary structure of WAS. We know that it doesn't compete with transferrin, and we know that it stains transferrin receptor expressing cells. But we did 5 rounds of sequential selection against these cells, and those cells also express other receptors that are internalized into the cell. So it is possible that despite the fact that we selected against the recombinant receptor, that we may have pulled something out that no longer binds transferrin receptor. So in order to confirm which receptor that we're binding, we did an siRNA knockdown assay. So basically, we take our transferrin receptor expressing cells, and we incubate it with an anti-transferrin receptor siRNA, which will result in a reduction in the expression levels of transferrin receptor. We then incubate those cells with fluorescently labeled Aptamer, and if we are specifically binding transferrin receptor, what we should see is a reduced amount of fluorescence in the TFR knockdown cells compared to control. If we are not specifically binding transferrin receptor, then the TFR knockdown would not actually show a difference to the control. And what we see when we actually did this experiment, by incubating cells with fluorescently labeled transferrin, C2, or WAS, is that we got, compared to the control, which is in green, we got a good knockdown with transferrin receptor transfection with all three molecules. And that's all just represented down here, indicating that knockdown of transferrin receptor results in a greater than 50% decrease in labeling by all three of these molecules, confirming the fact that they are all specific for transferrin receptor. So now we need a way to sort of compare C2 and WAS to each other, just so that Matt and I can really figure out whose aptamer is best. We already know mine is better because it doesn't compete with transferrin, but we also needed a way to see how well they bind to the transferrin receptor. So we did this by doing, we did a titration of the aptamers, incubating with cells, and then utilized the median fluorescence values of those FACS populations in order to figure out a half maximal binding constant, which we call an apparent KD. And what we found is that WAS and C2 have apparent KDs of 390 nanomolar and 140 nanomolar respectively. So this is the one point where I have to admit that Matt's aptamer is slightly better than mine. Okay, so now we know. We already know a lot that's going on here, and now we know that we're specifically binding transferrin receptor. The next question is WAS actually getting internalized with the receptor, or is it just being stuck to the surface? And this is important twofold. First of all, if we want to use this for therapeutic delivery applications, you actually need your therapeutic to be able to get into the cell. And second of all, since we know that transferrin receptor is constitutively internalized, if the binding of WAS is preventing its ability to be able to internalize into cells, then there's a serious implication that you could be having toxic effects from these aptamers down the road. So basically, all of our experiments thus far have just been looking at the total signal, and what we really need to do is to separate that signal into its component parts of internalized and surface-bound signal. And the way that we accomplish this was, in order to get the internalized signal, we incubate the cells with RiboShredder, which is just a proprietary RNase cocktail, and that will chew up any aptamers that are bound to the surface of the cell, so that just isolates the internalized signal. If we pre-incubate cells with deoxyglucose and sodium azide, then we can arrest clathrin-mediated endocytosis, and that will allow us to that will shut down the cell's ability to internalize transferrin receptor, and we will just be visualizing the aptamers that are able to bind to the surface. So after repeated apparent KD experiments on cells treated in this manner, what we find is that compared to untreated cells in blue, for both WAS and C2, RNase treatment had very little effect on the ability of the aptamer to internalize and left the KDs relatively unaltered. However, if we arrest the cells, we see an almost complete abolishment of the aptamer's ability to stain the cells, indicating that most of the signal that we are seeing in the untreated samples is primarily coming from the internalized signal. So WAS is able to get into the cells. And we can actually see that visually by doing a co-localization experiment, where we take cells and incubate them with an Alexaflor 488 labeled transferrin and a Tamra labeled C2 or WAS. And these are incubated on the same cells. So when you merge the two pictures together, what you see is the transferrin is in green here, and the aptamers are in red. And when it's merged, all of this yellow or sort of orange here is indication that these two fluorophores are existing in the same compartment, or are existing in near proximity to each other in the cells. And furthermore, by the fact that the image here shows a kind of punctate pattern, that gives us a pretty clear understanding that these two are existing within the endosomes together. So the last thing that we wanted to do to really have this complete knowledge of the characteristics of WAS is that we started this out with the hypothesis that C2 would not function very well in vivo due to its competition with transferrin. And so we tested this using an in vivo xenograft tumor localization assay. So basically what we do is we take cultured human 22RB1 prostate cancer cells, so these are human cells, and then we inject those into the flank of a mouse. 3 to 4 weeks later, the mouse will develop a human tumor on its flank. We then incubate our aptamers labeled with a near-infrared dye, and 12 hours later we take these mice and we live image them on an IVIS near-infrared imaging system. And this is an experiment that was performed by my colleague Dr. Lindsley Kelly, and what she found was that compared to C36, which is our control aptamer that should not be localizing to tumors and does not, C2 also does not really localize to tumors, confirming our suspicion that it's unable to localize to tumors in vivo. However, if we do this with WAS, we find good localization of the fluorophore on the tumors in 3 separate mice, indicating that WAS is able to localize itself to human tumors in vivo. So, now that we have a nice body of understanding of how WAS functions, we wanted to move ahead with one of our applications and find out if WAS could mediate the inhibition of New World mammarinovirus infection in vitro. So, from the previous set of studies, what we now know is that WAS does not compete with transferrin, but since we know that these viruses bind to the apical domain of the receptor, we need to actually figure out where it is binding, not where it isn't binding. And the hope is that WAS binds to the apical domain and that it is able to compete with transferrin. So the first thing that we did is hydrogen deuterium exchange mass spectrometry. And basically what you do is you take the recombinant protein and you flash incubate it in deuterium. And the deuterium will exchange onto the amide hydrogens of the peptide backbone, making those peptides heavier, so that when you digest them and submit them for mass spec, those peptides would be heavier than if you just digested the protein itself. If we do a follow-up experiment where you co-incubate the receptor with the aptamer, the theory is that the binding of the aptamer would protect the residues that it binds to from deuteration, thus leading to the peptides that are involved in binding having lighter masses, or a differential mass between the two experiments. So when we actually did this, if we graph out the change in mass between the two bodies of peptides, what we find is that while we get a scattering of change in masses for some of the peptides, one peptide in particular, number 49, shows a dramatic decreased change in mass, which indicates that this peptide was protected from deuteration by the addition of WAS. If we map that onto the crystal structure of transferrin receptor, we see that peptide 49 is actually located on the apical domain of the receptor, and in fact is only 15 angstroms away from the known binding site of the Machupo mammarinovirus. Additionally, this has also been shown to be the binding site of that apically binding anti-transferrin receptor antibody, clone MA712. So the mass spec data was a good start, but it can also have some problems. As you can see, there were other smaller changes in mass throughout the experiment, and it's known that binding of a molecule to a protein can result in distal conformational changes, which is why we see other peaks in that delta mass spec, the delta mass slide that I showed you. So we really wanted to back this up with a confirmatory experiment. And what we decided to do was use a competition internalization assay, similar to the way that we proved that C2 competes with transferrin, to prove that WAS binds to the apical domain. So the first thing that we did was we tried to see if that clone MA712 antibody would be able to prevent the internalization of WAS into transferrin receptor expressing cells. And what we found via flow cytometry experiments, which is just represented here in this bar graph, is that if you just stain cells with WAS by itself, you see a good signal, but if you compete it with that apically binding antibody, you see a 97% reduction in WAS's ability to internalize into cells. Compare that to C2, where if you compete it with this antibody, you see only a 7% reduction indicating that WAS is competing with this antibody for binding to the apical domain. We followed this up by expressing the machupo-glycoprotein, the one that facilitates the entry of machupovirus into human cells. We expressed this as an Fc domain and labeled it with a secondary antibody. And as we found out by flow cytometry, which is just represented in this bar graph, if you just incubate this construct with transferrin receptor expressing cells, seen here in this blue chart, if you compete it with WAS, you get an 87% reduction in internalization of that construct. However, if you compete it with C2, or our control aptamer C36, you see no effect on its ability to internalize. So, based on the HGX experiments, we were pretty confident that WAS was binding to the apical domain. And when we back that up with our competition experiments, we know that when we saturate the cells with WAS, we are able to prevent the binding of this machupo construct into cells. So now that we're pretty confident in its binding location, the big question is, will WAS be able to work against that machupo protein when it's in the context of the viral surface and be able to prevent viral infection of human cells? And one of the first limitations here is the fact that these viruses are fairly infectious and they're classified as BSL-4, meaning they need very rigid safety protocols when you're handling them. So in order to do our initial experiments where we just wanted something that could mimic the functionality of its entry mechanism, we decided to use a model system. So our collaborators developed this recombinant vesicular stomatitis virus, where basically you start with the VSV virus as a building block, and you recombinantly replace the glycoprotein of the VSV virus from its genome. You replace it with the GP protein, which expresses both the GP1 and the GP2 protein. You replace it with hunin or machupo, which are our two New World mammarinoviruses that bind transferrin receptor. And then as a control, we also use lossavirus, which is an Old World mammarinovirus that does not bind transferrin receptor for internalization. Our collaborators also inserted an EGFP expression construct into the genome, so that when these particles infect human cells, you actually get expression of EGFP. And this results in a BSL-2 level virus that we can work with here at Einstein that has its own infection reporter, but for all functional purposes, displays the same entry mechanisms as the wild-type viruses. So we take these viruses, and we do a viral inhibition assay, where we incubate these particles with human cells, and then if we get infection of those human cells, they will start to express EGFP, which can be visualized and quantified by microscopy. If we are able to impede the ability of these viruses to infect cells, then via microscopy we should see a reduction in the number of infected cells. And when my collaborator, Dr. Rohit Jengra, so the first thing that we wanted to do is see if we could recapitulate the ability of that clone MA712 antibody to prevent the infection of cells by these viruses. And what we found is that with the hunin virus and the machupo virus, this antibody was able to reduce the infectivity of these viruses compared to an isotype control, all the while having no effect on the lossavirus, which does not bind transferrin receptor, thus indicating that we were getting receptor-specific inhibition of viral entry. We now wanted to check to see if WAS would have this same functionality, and indeed, when we look at hunin and machupo virus, WAS was able to dose-dependently inhibit the virus's ability to infect cells, with EC50s of 400 nanomolar and 700 nanomolar respectively, all while having no effect on lossavirus, and just as you would expect, since C2 binds the transferrin binding site, it also has no effect on these viruses. So, for anyone who's done these experiments, or tried to do viral inhibition in general, you know that an EC50 of 400 nanomolar isn't that stellar. So we wanted to see if we could actually improve the ability of WAS to inhibit these viruses without making a new aptamer. And the way we wanted to go about doing that was to utilize avidity effects. So just to give you a really quick background, without getting into too much physical chemistry, The affinity, which I've talked about a lot in terms of like the apparent affinity of the aptamer for the receptor, is proportional to the enthalpy of binding minus the entropy lost by binding. So basically, when you have a receptor and the ligand binds to it, enthalpy is the amount of basically sticking together that you get, or the beneficial aspects of binding these two molecules together. And from that, you have to subtract the entropy, which is the loss of conformational freedom of this ligand from not being able to float around in solution anymore. Now, if you could imagine two ligands, so that gives you H minus delta S. If you have two ligands binding to two receptors, and you constrain them to each other with a non-flexible, perfectly spaced linker, what you can imagine is that the binding, you would have 2 delta H, because you now have two binding events, and when this molecule binds, it loses entropic value, and because of this inflexible linker, the second binding event is already there, so there is only 1 delta S worth of loss of conformational freedom. This isn't really practical, though, because we can't design perfectly inflexible, perfectly spaced linkers. And if you imagine that the linker is long enough, you would eventually have this thing flopping around so much that functionally it would be just the same as two molecules binding separately, meaning that you would have 2 delta S. So you wouldn't have the benefits of constraint. So the idea of harnessing avidity is to get entropically favored binding with an optimally lengthed linker without getting enthalpically hindered by preventing the optimal binding of those two molecules. So to test this, we wanted to make dimers of WAS using varying length PEG linkers. So we made constructs of WAS with a PEG 3, PEG 11, and PEG 22 linker, and then analyzed those for apparent KD. And what we found, which is just their KDs are just represented here in this bar graph, is that all constructs were better than WAS as a monomer. But the interesting thing we saw was progressively decreasing KDs as the linker got shorter, indicating that we were still getting entropic favorability by reducing the length of that linker, and we have yet to reach the point where we are actually enthalpically hindering binding. So this project is currently still ongoing, but we had to sort of take a deviation from it because we were having difficulty producing the large quantities necessary for doing our viral inhibition studies. So to get around our difficulty in producing it, we decided to sort of take a sidetrack and just use streptavidin-biotin interaction in order to make multimers. So basically what we did was we biotinylated our aptamers and incubated them with streptavidin, which will bind 4 biotins per protein, thus creating functional multimers. But the first thing that you need to check when you're making these things is whether or not the addition of this steric group on the end is going to affect the ability of WAS to function on its own. So when we made those, what we did was we just incubated biotinylated WAS with a 10-fold excess of streptavidin, and when we assessed the apparent KD again, we actually found that the addition of streptavidin to that molecule improved the affinity of WAS for the receptor. So this alleviates our concerns that it would hamper its ability to bind. We then went on to make our multimers. So what we did was we incubated increasing concentrations of biotinylated WAS with streptavidin, and as the ratio of WAS to streptavidin increased, we're hoping we would eventually make tetrameric molecules. But what we found is that we actually saturated streptavidin with aptamer at a ratio of 3 to 1, which is evidenced by the fact that we started seeing free aptamer in those samples. So we were actually functionally making probably a mixture of dimers and trimers, which we just termed multimers, presumably restricted by either steric effects or more likely the cost of trying to associate too much negative charge into one place and ending up with electrostatic repulsion. But we took these multimers and then assessed their affinity for cells. And just comparing to the data that I already showed you, indicating that C2 and WAS had their affinities of 140 and 390, when we assessed the affinity of our multimers, what we found is a dramatically increased affinity, roughly 30-fold, with C2 and WAS having KDs of 6 nanomolar and 12 nanomolar respectively as streptavidin multimers. So then we wanted to make sure that this steric molecule wouldn't be affecting our ability to inhibit viral infection. So we're just doing the viral infection assay again. And what we actually found was that on Hunin and Machupo, WAS had an EC50 of 200 and 140 nanomolar respectively. So we're actually seeing a twofold and a roughly fivefold improvement in EC50. So just having this streptavidin there as a bulky group hanging out on the backside of the aptamer is increasing our ability to prevent viral infection. And then finally, when we did this with our full multimer, we saw an even better increase in viral inhibition with EC50s of 30 and 70 nanomolar respectively, which relates to about a 10-fold improvement in our inhibition ability. The last interesting thing we saw here was that C2, when it exists as a multimer, actually manages to have an effectiveness against the Machupo virus with an EC50 of 200 nanomolar. And we hypothesized that this is probably due to the fact that the Machupo binding site is ever so slightly different from the Hunin binding site. So it's possible that by adding all of this extra sterics to the backside of the aptamer, it's able to reach out and block a portion of that epitope. So we now know that WAS is able to inhibit the infection of these VSV particles. And these VSV viruses are a really good model of viral entry, but they don't give you a really good indication of the virulence of the wild-type viruses. Because really all they're doing is they're sharing that glycoprotein complex. So the last thing that we wanted to do was to assess whether WAS would work on the wild-type virus. So we did this through a collaboration with John Dye at USAMRIID, and they assessed in just a pilot study if WAS could inhibit the wild-type Hunin virus's ability to infect human cells. And what we found is that compared to our C36 control aptamer, WAS did in fact have a dose-dependent inhibition of viral infection, approximately around 2 micromolar of an EC50, indicating that while WAS is not the best inhibitor of these wild-type viruses, this does show that we can actually use aptamers to inhibit the infectivity of these wild-type viruses. So that's everything I have to share with you today about WAS. But for as many questions that I've answered, there are a lot more questions that we have sort of brought up with this whole process. Some of the things that we want to look at in the future. So we know that transferrin receptor is upregulated in cancerous tissues, and we're wondering is, can we utilize WAS to specifically deliver chemotherapies to cancer? So the state of the art for chemotherapy up until a few years ago was basically you inject your body with poison, and since we know that cancer cells metabolize faster than normal cells, then those cancer cells will metabolize the poison, the toxins, faster. But if we include the concept of the fact that these cells with faster metabolism are actually expressing higher levels of transferrin receptor, then we can conjugate toxic cargoes to transferrin receptor ligands in order to increase the specific delivery of chemotherapies into cells. So Christina Krashmer in my lab has actually started the process of conjugating toxic cargoes to WAS to see if we can deliver them specifically to cancer cells. The other thing that we wanted to look at was we know that transferrin receptor is expressed on the surface of the blood-brain barrier. So the question is, can we actually use WAS to deliver molecules to this privileged space on the other side of the blood-brain barrier? And so basically, the blood-brain barrier is the vasculature in the brain that delivers all of the blood and nutrients. And it has very restricted properties on what's able to cross it because it's the only line of defense keeping toxins out of your brain. The state of the art is basically sticking a needle into your brain and injecting the therapy where you need it. But the vasculature actually provides a very intricate delivery system if you can manage to find a way to harness it, because it is so pervasive that no cell in your brain is actually more than 25 microns away from a blood source. So if we could utilize this system to deliver drugs, that would be advantageous. And it just so happens that transferrin receptor is able to transcytose across the blood-brain barrier to deliver nutritional iron to brain cells. So some groups have previously used transferrin or transferrin receptor antibodies to deliver diagnostics or therapeutics to the brain. So we are currently under undertaking the process of finding out if we can utilize WAS in a similar manner to use an aptamer to actually deliver things to the brain. Finally, in the interest of competition selection as well as viral inhibition, we learned a lot of things about selection through this process. First of all, we know that internalization selects rewards cell accumulation over affinity, because you're actually going to enrich something more if it binds to the receptor, gets internalized, and then falls off once it's in there so that it stays in there. So, if we want to actually be inhibiting viruses, we found from our avidity studies that a higher affinity is better. So we would like to redo the selection entirely on recombinant transferrin receptor, but utilizing that competition selection to see if we can find higher affinity variants of WAS. We would also like to use new enzymes that have been developed in the last couple of years so that we can increase the nuclease stability of our aptamer library, utilizing a 2'-O methyl library that would greatly reduce its nuclease susceptibility as well as immunogenicity. And then finally, if we're going to continue with these viral studies, one of the detriments to WAS is that we found that it is exquisitely human-specific. So if we wanted to move into preclinical studies, we would need something that would be able to function in non-human primates. And to do this, we theorized that we could use a toggle selection that switches back and forth between human and monkey transferrin receptor expressing cells to see if we can find a molecule that is cross-reactive. So with that, I have a number of people I need to thank because I stood on the shoulders of many giants throughout this process. The first of which would be my mentor, Matt Levy. He is the incarnation of everything that you would hope for in a PI. He is brilliant. He knows everything. He's always available to help. He is a fountain of ideas. One of the bad things is that Matt is a fountain of ideas. He comes in the morning, and you can just see his tail wagging because he is so excited about these 5 new projects that he thought of. And I'm going to go and pilot all of them real quick, just on the side, while I'm still working on all this other stuff. But I actually love that because I really like messing around with new things and just finding out if something works and if it's somewhere that we should actually go. But it's been a lot of fun, and I really appreciate everything that you've done. The man behind the curtain, Amos, for everything that is amazing about our lab, Amos has a hand in it. She makes sure everything runs. She makes sure that there's food and beer at every one of our lab meetings, which makes us the jealousy of every other lab on campus. And she is just a fantastic and loving person to be around. I have to thank my entire lab. That's why I took all this space to thank them. They are such a supportive group. They really make coming to work a great thing, even on those days where you don't want to come to work. Marcos, Arjit, Francine, Lindsley for listening to me all the time while I had to complain about stuff. Paul for taking me rock climbing all the time. Christina and Nachi, good luck without all of us there. Lulu, Brian, Simon, Sinald, Way, Drew, and of course Samantha Pants. She's my partner in crime from the beginning. And of course, if I'm going to have to thank all of my lab members, I have to thank Sarah Hanafar for being our lab pet. We kind of adopted her. It was adorable. Scientifically, Dr. Kartik Chandran and the Chandran Lab particularly wrote HIT for all of the work that they did with the viruses. I basically read one paper that said these viruses bind transferrin receptor, got really excited. Matt set up the meeting, and then they kind of carried us on their shoulders and really brought us into an entire field that Matt and I both had no clue about. Probably still don't. Eric Snap for doing the microscopy with me. I really appreciated it. Hui Zhao for doing the HDX experiments. Aaron Golden for lending us Kevin, because no one still knows how to analyze deep sequencing data. But Kevin is, if you ever need something, Kevin is the guy to come to, because he can analyze that data for you. And for all the stuff that I talked about, there's probably an equally expansive amount of work that I didn't talk about, mostly because it didn't work, or we haven't finished it yet. We have started with the trying to deliver WAS across the blood-brain barrier, and Dr. Prasad, especially Vasudev Rao, were helping us set up that blood-brain barrier transcytosis model. We also tried to get a crystal structure of WAS and transferrin together, and Dr. Scott Garforth in the protein production facility made us gobs of protein, which is not a trivial thing to do. And then Raf Toro and Vladimir helped us set up the crystal trays, which we got crystals after 24 hours, but they didn't have a good diffraction, so more science. But that work does still not go unrecognized and was still greatly appreciated. Our collaborator at US AMRID for doing the live virus stuff. And then my committee meeting. I've been fortunate to have them stick with me through the entire process, and they all stuck on to be on my thesis committee. So Drs. Kartik, Steve, John, Vinyaka, I appreciate all of you for helping steer Matt and I through all of Matt's ideas that he wanted me to do, and coming out with a cohesive thesis in the end. Also Debbie for agreeing to be my outside examiner and driving down here from Boston to be with us today. And also Ian for agreeing to be my backup just in case. The department doesn't run without the biochemistry office, and all of those people are deeply appreciated, and also everyone in the graduate office for helping us out. And finally, I just need to acknowledge the funding that made all of this possible, which came from the Stand Up to Cancer, as well as our support from the NIH. And with that, I can take your questions.
Aptamer also be taken up
into cells? We didn't actually do the like arresting cells and RNase degradation, but our theory is that it does, because, well, so our theory that our sort of supporting theory for that they're getting internalized is the fact that it appears that these aptamers have really high off rates, and while the multimerization probably had a significant improvement of the off rate, there's still the matter of there's washing and getting it all the way to the fax machine before it finally gets imaged. So it should definitely be tested, but my theory is that they are getting internalized. Because we did some work with 200 nanometer nanoparticles, and we saw by microscopy that those could get in, so I would theorize that the strip tabbing is as well.
Do you have any left over that you can give to me?
Yes, I've had your email. I had a couple of things to prepare for this week, but...
Last question. Did you brew any beer for tonight's events?
No. It was on my to-do list. I wish I had.
Any other questions? Yeah.
Why do you think that the adenovirus showed a higher affinity than the more toxic viruses, or that why your Aptamer combination was more effective against the adeno than against
the wild type? Like the wild type versus the VSV?
Or the June
Yeah,
virus?
well, so it was the wild type quinine, and then there was also the recombinant VSV modified to express quinine glycoprotein. So our collaborators at US AMRID tell us that, well, so first of all, I mean, we're recombinantly expressing that glycoprotein on the surface of a virus that isn't expecting it, so it kind of, those viruses are a little hampered. And also the wild type viruses are just better at infecting cells. Because we're only modeling the entry mechanism with the recombinant virus, and they're all the downstream parts of viral life cycle, and those viruses are just better than the VSV, from my understanding. I'm going to take one more minute then to thank a couple more people. I will be brief, I promise. So, real quick, the first thing I need to do is to thank all of my people at Einstein. They really should put it on the website, on the front page of the recruitment page, that the people here are just so great. And I started going through and trying to find pictures of everyone that I wanted to cover, and I was preparing for my thesis, and it was getting really stressful, and I was like, I'm going to leave someone off. So instead, I decided the easier way to go about this was just to tell off the people that I didn't like, so that the rest of you know that I appreciate you. So I'm going to have my no thank yous now. So I started going through Facebook, finding all pictures of all those people, and it turned out easier than I imagined, because really, like, everyone at Einstein has been fantastic. And I don't have your picture up here right now, because I wanted to make sure that you all know that I think you are great. And hopefully over the course of the last 6 years, I've proven that to you with the way that I've treated you and interacted with you. I need to thank my family. That picture did not turn out, but I assure you they're real people. Extended family. I have a lot of people here in the audience today. My parents-in-law, my parents, step-parents, step-aunt. I've got my cousin here. I've got a lot of people. I got a lot of people who wanted to be here but couldn't, but they're possibly watching on the live stream right now. So thank you to everyone who tuned in. And then finally, there's just one woman that I really need to thank, who will probably murder me if I don't. Kate has been a fantastic support through all of this. Right from our first date, she decided she liked me, even though I'm a bit of a weirdo. Something that I have not grown out of. But she always keeps a smile on her face and is very supportive and loves me unconditionally, even when I'm not that great of a person to be around. We've climbed mountains together. We have run the gauntlet together. A lot of you probably don't recognize her from these photos because you only see the product of her generosity, which is anyone who's ever come within 10 feet of me at Einstein has received some of her baked goods or something that she made. I mean, heck, she cooked dinners for everyone for at least 20 people in my class for 5 nights in a row the week that we were all having qual. So everyone just came over to our place and she took care of everyone. And we all owe her a debt of gratitude. And yeah, she's fantastic, even if she's a little weird too. But we don't know where we're going next, but we know that we've enjoyed our time here and the time that we've spent together. And I look forward to figuring out what we do next. So, thank you for listening. Sorry I ran over.